Hello students, I am Novelty Fatima May M. Giang, your student teacher intern, and welcome to General Biology 2 class. In your previous lessons, you have discussed that genes are the materials responsible for the traits that every organism inherits, possesses, and passes on. You have also understand that a gene is a portion of a chromosome carrying information needed for the synthesis of proteins, one of the major building blocks of life. But before we go on to this lesson, let's have first a brief definition of what is molecular biology. Molecular biology is a branch of biology that deals with the molecular basis of biological activity. This field overlaps with other areas of biology and chemistry, particularly genetics and biochemistry. As we all know, the deoxyribonucleic acid or DNA is the material that carries the genetic information of organisms. Before this was confirmed, many biologists were studying the tiny interior features of chromosomes in order to locate the main source of genes. A British bacteriologist named Frederick Griffith was one of the first scientists to advance the hypothesis that DNA carries genetic material. He achieved this through an experiment involving the transformation of the bacterium, namely the Streptococcus pneumoniae, when injected in mice. There are two major forms of this bacterium: the smooth strain, or the S strain, and rough strain, or the R strain. The S strain bacteria contain a protective capsule and can cause diseases, thus killing the mice a few days after injection. On the other hand, R strain bacteria lack protective capsules and do not kill the mice after injection. Griffith also used a heat-killed S strain in the experiment. He used both live and heat-killed bacteria to see how they would affect the mice. As you can see in the figure, R strain bacteria and heat-killed S strain bacteria did not kill the mice after injection. On the other hand, live S strain bacteria and the combination of heat-killed S strain and R strain killed the mice. When Griffith isolated the combined heat-killed S strain and R strain bacteria from the diseased mice, he found out that R strain had been transformed into a virulent S strain bacteria. From these observations, Griffith hypothesized that a certain chemical component or transforming factor within the virulent S strain bacteria might have transformed the genetic composition on the R strain and made them disease-causing type of bacteria. The group of Oswald Avery, Colin McLeod, and Maclean McCarthy had been investigating pneumococcal infections when Griffith published his results. These three scientists looked into and replicated Griffith's experiment in vitro but applied modifications. They extracted materials from the S strain cell for the investigation and added enzyme treatments to destroy any DNA, RNA, or protein by combining the mixture with live R strain bacteria. The group of that found out that R strains transformed S strains, except in solutions where DNA was destroyed. This confirmed that the transformative factor of the S strain in the DNA, implying that it contains genetic material. The hypothesis was further confirmed when Martha Chase and Alfred Hershey worked on bacteriophages, viruses that infect bacteria. They found out that DNA is the genetic material of the T2 virus, which infects Escherichia coli or the E. coli bacteria. Chase and Hershey used the T2 bacteriophage because its the main components are DNA and protein only, and it can remodel its host cell to produce new phages. The scientists just needed to figure out whether it is DNA or protein that enables this process. Chase and Hershey's experiment involved radioactive sulfur solutions and radioactive phosphorus solutions, which they used to identify DNA from protein. Chase and Hershey knew fully well that phage proteins contain a lot of sulfur, while phage DNA contains phosphorus that is absent in proteins. After the infection, the mixture of bacteria and bacteriophage was centrifuged at a high speed to form layers. with the phage on top and the bacterial forming the pellet at the bottom of the test tube. They measured the radioactivity of the layers and found out that the radioactivity of the proteins were suspended in the liquid, suggesting that protein is not capable of entering the cells. On the other hand, the radioactivity of the phage DNA infecting the bacteria was located at the pellet of the test tube. When they destroyed and opened the bacterial cells, they found out that the new phages carrying only radioactive phosphorus in their DNA were detected, confirming that DNA is indeed the genetic material. Now that you already know about the history and the early studies that says DNA is a genetic material, let us now proceed to the structure of DNA. The DNA is a complex double helix ladder made up of interlinking nucleotide subunits, a deoxyribose sugar molecule and a phosphate group, and nitrogen containing bases that make up the ranks of the ladder structure. The nitrogenous bases are further subdivided into purines and pyrimidines. When we say purines, consist of guanine and adenine, while the pyrimidines consist of thymine and cytosine. There were a number of scientists involved in the discovery of the structure described previously. We have Erwin Chargaff, who first analyzed the nitrogenous bases of DNA, establishing two rules about the way these pair up. According to Chargaff's first rule, in any species, DNA follow a particular genetic pattern in which the amount of units in one purine nearly equals the amount of units in one pyrimidine. Keeping the following pattern, adenine is equal or nearly equals the amount of unit of thymine, and guanine nearly equals to the amount of cytosine. The second rule states that the amount of each base of adenine, thymine, guanine, and cytosine in the DNA varies from the species to species. Moreover, the double helix structure of DNA was confirmed through the X-ray crystallography experiment of Rosalind Franklin and Maurice Wilkins, who pointed X-ray beams at the DNA sample to reveal its form. Chargaff's rules and Franklin's X-ray diffraction patterns contributed immensely to James Watson and Francis Crick's theory about the DNA structure. So in 1953, the scientists had their study on DNA published in the British scientific journal Nature, which featured their DNA structure model. This model presented a three-dimensional double helix structure with the sugar phosphate groups on the sides and the base pairs on the inside. Crick and Watson's model was consistent with the descriptions given earlier by fellow scientists, including Chargaff and Franklin. For this discovery, they received the Nobel Prize for Physiology in 1962. Now, let me ask you, how did DNA and RNA differ from each other? While both DNA and RNA are used to store genetic information, there are clear differences between them. This table summarizes the key points. And that ends our lesson for this day. Thank you for listening, and please don't hesitate to ask questions.